In this session, we'll learn how to use bioinformatics applications to predict protein structure. The best way to study protein structure is to use a three-dimensional structure from X-ray crystallography. Unfortunately, only a modest subset of proteins have a 3D structure available. Therefore, we're left to try to predict protein structure based on information in the sequence of the protein. Using well-established algorithms, you can make reasonable predictions about whether a part of the protein is likely to be a alpha helix or a beta sheet, whether the region is likely to be hydrophobic or hydrophilic, what the probability of a particular area appearing on the surface of the protein is, whether a region is likely to be transmembrane, as well as the ability to predict the antigenic properties of certain parts of the protein. None of these algorithms are flawless, and you must, in many cases, look at multiple algorithms to see if there's agreement between them or not. In this session, we'll use the Protein 3D application, a module within the DNA Star commercial bioinformatics suite that has been provided under an educational license. So let's open the Protein 3D module, and then within that we'll open the CFTR protein file that we created in an earlier exercise. As soon as we open it, we'll see a very rich visual environment for Protein 3D. The pane on the right allows you to determine which of the analytical algorithms you're viewing. So let's view a few additional secondary structure predictions. We'll add Garnier-Robinson, for example, and one other feature. Now let's go over to the visualizations and see what we've got to work with. Starting from the top, we can see the algorithms that predict whether a region is likely to be an alpha helix or a beta sheet. So in the green, we see the regions that are likely to be a helix, and in the yellow, a beta sheet. A nice feature is that the entire protein sequence is displayed below, and as you select regions above, the sequence that you've chosen are highlighted in the pane below. So that region is predicted to be a beta sheet, and so is this shorter region next to it. And so you can see that as we select features above, the sequences are shown below. Let's go to Analysis and choose to zoom in a little bit closer so we can get a better view of the region that we're working with. One thing you notice is regions predicted to be a beta sheet are not predicted to be an alpha helix, and this will hold true across most of the secondary structure prediction algorithms. This is an antigenicity prediction. We're not going to focus on that too much in this session. This is one algorithm, the Kyte-Doolittle algorithm, for predicting whether a region is hydrophobic or hydrophilic. And again, as we noted above, a region that's predicted to be hydrophobic won't show up in the hydrophilic predictions. Protein 3D offers a couple of algorithms. So Hopwood's is another algorithm for predicting hydrophobicity. It also offers algorithms to predict the likely charge of a region, and many other algorithms that are useful for understanding the predicted protein structure. In this session, we've seen some basics about using a bioinformatics application to predict the secondary structure of proteins based entirely on the protein sequence.